Hello everyone, I am Shreyoshi Das and we are here to perform a very simple experiment which involves gram staining or cell wall staining. We are here to understand the basic differentiation between the chemical and theoretical aspects of gram staining, as well as to differentiate between the two groups of bacteria, that is gram positive and gram negative. Gram staining was introduced to us by a Danish scientist called Christian Gram in the 1880s, and it is one of the most commonly used methodologies to differentiate between the two groups of bacteria, gram positive and gram negative. The basic difference that lies between these two are in the structure of their cell wall. The gram positive bacteria have a very thick cell wall, usually made up of 60 to 90% of peptidoglycan. It also contains teichoic acids, which gives a positive charge to this bacteria. This bacteria retains the primary stain and remains purple in color, whereas gram-negative bacteria have a very thin cell wall, usually made up of 10 to 20% of peptidoglycan. It also contains an outer membrane, which is usually made up of lipopolysaccharides and gives a negative charge to this bacteria. They lose the primary stain and takes up the counterstains, safranin, and turns pink in color. There are 4 chemical reagents that we use in this experiment. First is the primary stain, for which we use crystal violet. Crystal violet stains all the bacteria purple in color. Then we have an iodine solution. Iodine is added to fix the stain inside the bacteria. It also forms a complex with the crystal violet called CV1 complex and remains inside the bacteria. Next, we have 95% ethyl alcohol, which is used as a decolorization. Alcohol decolorizes some of the bacteria. The gram-negative bacteria loses its outer membrane, creating tiny pores which allow the CV1 complex to leave out of the bacteria. Whereas gram-positive, because of their thick cell wall, they do not lose the CV1 complex and retain the CV1 complex. The last, we have a counter stain, which is safranin. It stains the gram-negative bacteria pink in color because of the tiny pores created due to the loss of the outer membrane. Whereas the gram-positive bacteria still remains purple in color because of their thick cell wall. So now we move to the materials that are required for this experiment. The 4 chemical reagents that we require are crystal violet, iodine solution, 95% ethyl alcohol, and safranin. We also require 18 to 24 hours old nutrient broth. The equipments that we require are inoculating loop, staining tray, glass slides, blotting paper, and microscope. So now under sterile conditions, we start the experiment with first wiping our hands with alcohol, taking some amount of the blotting paper, cleaning the slides with alcohol on both the sides. Through this. Then we mark a circle on the slide. Turn it to the opposite side, and it is here where we'll make the smear. We take the inoculum tube, this is our overnight broth, and take some amount of it with the inoculum tube and make a smear on the circle. We do it in a circular motion. Now we close it and keep it aside. we on the gas flame. And we now need to heat fix this slide. We normally do it from the upper side of the flame and on the bottom portion of the slide. Our slide is now fixed. We off the flame. And now we move to the following procedures. Now we put the slide on a staining tray and we add our first primary stain, which is crystal violet. It will stain all the bacteria purple in color. We add a few drops to it and leave it undisturbed for 1 minute. So now our 1 minute is over and we wash the slide with distilled water. We pour the water around the smear and not on the smear. Next we move forward to our second stain, which is the iodine stain. The iodine will form a complex with the crystal violet called CV1 complex. We leave the iodine solution undisturbed for 1 minute. So now our 1 minute is over and we wash the slide with distilled water. And then we move forward to our third decolorizing agent, for which we use 95% ethyl alcohol. The alcohol is added to the smear dropwise. We wash it off with the distilled water to ensure even decolorizing. And then we move forward towards our fourth stain, for which we use Safranin. It is our counter stain. We add few drops of the Safranin on the smear and let it undisturbed for 1 minute. After 1 minute is over, we wash off the stain with distilled water. And now we use a blotting paper to remove excess of water around the smear and let the slide air dry until the water is evaporated. Then we observe the slide under a microscope. Now we are going to observe the slide under the microscope. We adjust the slide and then we move forward to observe it under the 40x. And then finally, for a clear vision, we observe it under 100x. Before observing it under 100x, we put a drop of oil on the center of the slide and adjust it to 100x and then observe it under 100x. Now we have a clear vision of the two bacteria, gram-positive and gram-negative. The gram-positive bacteria can be seen stained as purple, whereas gram-negative has taken up the counterstain Safranin and has showed a new color pink. This is how we differentiate between the two bacteria, gram-positive and gram-negative. I hope the video was informative. We'll now next meet in the upcoming video. Thank you.